Excellent. Shall we start? Excellent. Oh, that's a good mic, isn't it? Hey, welcome to this event, Making Cancer Treatment Worth It. My name is Natasha Mitchell. I'm a presenter at the ABC. I present a show called Big Ideas, which goes out nightly on ABC Radio National, which in Adelaide is 7 to 9 a.m. But of course, you can get Big Ideas on the ABC Listen app. So if you haven't downloaded... Oh, no, start again. Right. Yes, okay, all right. Yes, I'm used to having mics up close. So Big Ideas, you can find it on the ABC Listen app, and we are recording today's event. I'm really thrilled to be joining you here on Kaurna Country, the first doctors, the first keepers of medical knowledge, the first scientists, over 60,000 years of history and knowledge keeping and sharing and making. I want to also welcome you if you're joining us on the live stream. Thank you so much for logging in and being with us today for this event here in Adelaide on Kaurna Country. And we'd love to involve you with your questions. I'll explain how to do that later. We'd love to involve you in the room here at the University of Adelaide with your questions and comments as well. But we'll have a bit of a chat with these incredible researchers, scientists, and then I'll open it out to the room and also to the live stream. Our topic today, making cancer treatment worth it. Cancer treatment can and does save lives, and some of you in the room and on the live stream may have had experience of cancer treatment, either your own experience or that of a loved one or a friend or a colleague. Sometimes we have cancer treatments that have horrendous side effects as well. So it's a real toss-up sometimes. What is that tension between saving your life and the quality of your life, especially when you're going through treatment? And also there's the cost and availability and accessibility of treatments. And today we're also interested in really connecting with new frontiers in cancer treatment and cancer research. It so happens that we have a stack of leading scientists in Adelaide this week for their big annual scientific meeting through the Australasian Society of Stem Cell Research. Some are working to stop cancer cells in their tracks, and they're using their knowledge around stem cell science to do that, or to understand how that might be possible. So we're stealing some of those scientists today for this event, effectively. And I want to thank our co-hosts here at the University of Adelaide South Australian Immunogenomics Cancer Institute, which is right here in this building. They're doing trailblazing work on cancer immunotherapy. They're also sequencing the genomes of people who are living with cancer, but also the tumours that they have, to really try and understand how we might personalise the medical treatment that people living with cancer receive. So thank you for hosting us. And we also have the National Stem Cell Foundation of Australia supporting this event, and thank you for the work that the foundation does in supporting stem cell science here in Australia. So to participate, when we come to you, we'll have a roving mic. It'll be in my hand. I'll come to you with the mic. Don't be shy. No question is a dumb question. You know, we've all been affected by cancer in some way, so we're learning together today. So really take this opportunity to ask whatever question you have. And if you're on the live stream, this is what I'm going to get you to do. I'm going to get you to email Tanya at scienceinpublic, all one word, .com.au. I'm going to give you that email again later, but it's Tanya, Tanya, at scienceinpublic, all one word, .com.au. Let me introduce our esteemed panel. Over on my far right here, we've got Professor Mark Dawson. He's a haematologist. He's a clinician-scientist at the Peter MacCallum Cancer Centre and at the University of Melbourne. His work has revolutionised, and that's a big word, but it's the truth, our understanding and treatment of blood cancers like leukaemia. He's also Sir Edward Dunlop Fellow for the Cancer Council of Victoria and was awarded Life Scientist of the Year at the PM's Prize for Science. Let's give him a warm welcome. We've got Associate Professor David Elliott. He's a stem cell researcher specialising in that beautiful organ inside us, the heart, our wonderful heart. I love my heart. In understanding how it develops and what can go wrong with it. He leads a group over at the Murdoch Children's Research Institute and is a principal investigator at the Novo Nordisk Foundation Centre for Stem Cell Medicine. And he is relevant to this conversation because he's doing really important work to try and minimise the impacts of chemo, particularly on children who experience leukaemia. So chemo can have some really difficult impacts. Yes, it saves lives, but it has a whole lot of side effects. We'll find out more about that. Let's give him a warm welcome. We've also got Professor Megan Munsey. She's a scientist focused on stem cell ethics and education at the Murdoch Children's Research Institute. She happens to be the president of the Australasian Society for Stem Cell Research, and she's also at the University of Melbourne. Megan does really life-saving work, and that work is helping us not seek out dodgy, unproven stem cell therapies, which is a massive marketplace around the world, including to some extent here in Australia. But Megan's work has helped change reduce the risks that people experience when they go to a private stem cell clinic claiming to be able to fix them and cure them of all sorts of things. It's her work at the national level and the international level to try and regulate those clinics that is helping save our lives. Welcome, Megan. Mark, I'll start with you. What makes cancer so especially tricky to treat? It's a big question, but it's an important question.
It's a big question, Natasha. So firstly, thank you everyone for coming out here in person and for tuning in online to this important discussion. Cancer is not a single entity. You know, it's not a uniform beast, so to speak. And we'll never get to the point where there is a single treatment for all cancers. By its very nature, cancers change and adapt to whatever the pressure is that we're putting on them. And it is that evolving nature of cancer that makes it really tricky to treat.
Yeah. Let's drill into that a bit further though. For example, individuals respond quite differently to a given treatment regime. Can you just give us a sense of what's going on at the cellular level, at the genetic level, that makes it so variable?
So we've known for decades that two different individuals that have a cancer that looks the same under the microscope will respond very differently to treatments. And in part that is because we have to characterise this cancer in greater depth beyond just looking at what it looks like underneath the microscope. More recently, what we've learnt is that even in a particular person, when we biopsy a cancer, that is made up of a mesh of cells, each with a different genome, different DNA mutations, and different consequences of those DNA mutations that gives you further heterogeneity. So we have two types of heterogeneity. One is interpersonal heterogeneity, which is the differences between two individuals that may have a similar type of cancer. And another is called intra-tumour heterogeneity, which is the differences in the cancer cells at the cellular level in the same person.
Yeah. So when we say someone's got lung cancer or a blood cancer, it's not one entity, it's not one thing.
Absolutely not. And also we've got to appreciate that we're classifying these things at a snapshot in time, and it's the minute you start to put pressure on this cancer by treating it, it'll evolve quite separately in different areas.
Yeah, now that's very interesting, and that's a great interest to you, isn't it? But cancer treatments still make people very sick. The treatment itself makes people very nauseous. Their hair fall out, radiation burns. It's a really tough slog. And often it's not very it's quite not very well targeted, is it? It affects the entire body. And all the systems in the body to an extent. So how would you describe how things have changed over the last 10 to 20 years in cancer treatment?
So I often explain it to my patients in the forms of pillars, the pillars of cancer treatment. For centuries even, we've had surgery, which was probably the first pillar of cancer treatment, being able to cut out things that we saw was abnormal. Over the last 50 to 70 years, we've had radiation and chemotherapy, and they've largely been aimed at cells that are dividing very rapidly, but they're indiscriminate in their activity. That means it'll target not just rapidly dividing cancer cells, but rapidly dividing normal cells, the cells that line your gut, giving you nausea, diarrhea, the cells within your bone marrow that produce trillions of cells each week for your blood cells. Right? And so you have low blood counts as a consequence of that. You know, the cells that help your hair grow, which is why your hair falls out. Right? So that's conventional chemo and radiotherapy. And
still very much a mainstay.
Very much a mainstay in a number of cancers because it's effective. Right? And so, you know, until we get better treatments, it'll continue to be a mainstay. What we've had over the last few decades is greater insights into what drives a cancer. Right? And so we've been able to build better targeted therapies to target a particular gene that might be driving a cancer. We've also developed immunotherapies, the ability to license your own immune system to go and see this cancer as being foreign and attack and kill it. So those are the evolving pillars of cancer therapy that are making things, in part, less toxic, more tailored in a personalised way.
We'll come back to some of the other new frontiers that will improve what's there even further. David, what about you? So you're a heart stem cell scientist, and what got you interested in looking at the impact of, say, chemotherapy, particularly on young people, on children who, you know, it must be a tough decision for parents. You want to save your child's life, but you're also potentially impacting their health for the rest of their life. Paint that picture for us.
Thanks, Natasha, and thanks everyone for having us. Yeah, as you touched on, it's very challenging for the families, and I think when I moved to the kids' hospital in 2013, we moved because the stem cell technology got to a point where we could, you know, effectively recreate the patient in the dish, and that meant that we had a system where we could look in and look to those cells and look at those heart cells and say, you know, why is about 20% of children who show up to be treated by clinicians like Mark, why do about 20% of them have heart damage? And then about 3% of them leads to heart failure and they need a transplant. So strokes,
heart failure, heart attacks,
things like that. And particularly, we really, many of our children survive. The success of the cancer field has been amazing. About 85% survive now. But then they've got to go and see another doctor because they've got to go and see their cardiologist. And the other problem we have, which will shock many of you in the room, is teenagers don't listen to their parents and they transition to adulthood and they stop going to their cardiologist and then they reappear in their mid-30s. So if we can stop it at the source, if we can say those drugs you were talking about that cause hair loss, and Mark was saying they hit the cells that divide, heart cells don't divide. So you don't get them back. So if you damage them once, that's it, they're gone.
Let's just go back though. Can you just describe what damage chemo does to the heart?
Yeah, so again, it's one of those things we don't understand that much. So there's guesses. We think that maybe some people, their blood vessels might collapse and their heart cells sort of die. I think some people, their muscle cells are just more susceptible. We don't know why. It's that, the differences amongst us all. Some patients are sensitive and some patients are resistant, and we are trying to dig into that and work out whether there's a genetic cause or what the actual cause is. So the damage is essentially, the same as, the treatment is to kill a cell. That's why you're trying to kill a cancer cell. It's just that you're killing your body's own cells. So, yeah. And then the downstream of that is if you get heart failure, it's because we kill too many heart muscle cells. So can we try and refine it so that we don't do that, or can we find ways that stop that from happening?
But it's really significant, the impact, isn't it? What was the 15 times more likely to have...
a
Heart failure.
Heart failure, yeah, than the general population. So it's quite high. And then there's the later one. So you either get patients who it happens very acutely, on treatment, and then you'll get some about 6 to 12 months later, and then some a bit further along. So, but compared to an untreated population, it's much higher, much more significant, and that's why part of your treatment regimes now is you'll see a cardiologist before you start, and then they'll monitor your heart health as you go through your treatments regimes.
Yeah. So it's a huge issue, and you've developed this model as a stem cell scientist of effectively a heart in a dish. So what are you doing with that heart in a dish to try and ameliorate the impacts of chemo?
Yep, it's a good question, and probably a crazy idea. But so the idea is that can we just recreate that damage in the laboratory, and then can we find other drugs or other treatments that stop it from happening? So if you're a sensitive patient downstairs in the clinic, in our hospital, downstairs in the clinic, if we take some cells from you and turn them into heart cells in the lab, you'll be sensitive in the lab. If you weren't, you won't be. So now we've already got an automatic distinguisher. And then we can look at and say, well, what's the difference between a sensitive individual and a not sensitive individual? And then we can say, well, can we find other things that we can put on the cells that stop the damage?
So not everyone is vulnerable to this impact on the heart. So you've got to determine, okay, what is it about individuals that make them more sensitive? And you've actually managed to find out, to some extent, what makes some people more vulnerable to having their heart damaged. So,
I mean, we have some clues. I think we're at the early stage where we're using, DNA sequencing technology and being able to say this person had a particular genetic change and they were sensitive, whereas this person doesn't. And then we can correct that genetic change in the lab and confirm that. So we're sort of at the correcting stage, but we've got a few clues about how it's happening.
But you've gone one step further, haven't you? You've thrown all sorts of possible drug candidates,
yes.
chemicals,
yes, a lot of
at
chemicals.
These hearts, poor little heart cells in a dish, stem cells, and you've found some possible concoctions.
Yeah, so again, these are all early, very, very early stages. What we have done is we've ruled out pretty much all of the compounds that are available at the moment. So all of the approved drugs, none of them really protected the heart.
Wow.
So we can eliminate those.
So kids were being given those drugs?
No, these are just every drug that's available to mankind that's approved by the FDA. So whatever it is.
Oh, you just tried them all. Right.
And then we said, well, they don't work.
It's so interesting hearing how science gets done.
Then we needed some new ones. So then we said, well, let's just look at some new ones. And so far we've done around 60,000 new ones, which is not many.
What?
Yeah, it's not many. Big Pharma will do millions. So it's a start. And, we've got some promising things, but there's still, anyone who's been involved in developing new therapy or a new drug, it's a 10-year process.
Yeah. I want to come back to that because I know that for people who are in the community of patients who are living with cancer, one of the great frustrations is getting access to treatments, getting access to clinical trials, getting results from the lab. I mean, people I know who've lived with cancer, they know the science really well, and they know where all the new frontiers are, and they are tracking, whether things are landing in the lab quick enough, from the lab into the clinical trials quick enough. And I'll love your advice, your thoughts about that, Mark, in a moment. But Megan Munsey, so the question of whether treatments are adequate or whether they have nasty side effects is a big issue in the stem cell field, isn't it? Because what you've been tracking is where people turn to for alternative therapies and unproven therapies, and particularly therapies that are called stem cell therapies. Just tell us about your interest in that, and then we'll consider the cancer question.
So thanks, Natasha, and thank you for coming along today. It's
Pleasure.
wonderful to have you here. I think that the challenge that we've long been interested in is this kind of co-option of the excitement and the promise of stem cell science, and the fact that we already use stem cells, as Mark has mentioned, to treat certain types of cancers, to to treat problems in the blood and immune system. But what we've seen for now decades, unfortunately, is enthusiasts who kind of use that excitement in the community and start offering something that has no testing. So you've heard how Dave is carefully going through and testing thousands and thousands of potential drugs that could help, particularly children in this case, who are facing chemotherapy. But what if you're diagnosed with another condition? I mean, the first thing many of us do is go online.
Yeah.
And there you'll find clinics that are, and doctors and companies who are basically saying, come to me now, because you don't need to wait. You don't need to wait for that testing. We have already treated thousands of patients like you. You just need to bring your checkbook. And it's that, I think, misinformation and exploitative marketing that has really sort of driven me and my interests and many of my colleagues around the world to kind of monitor what's happening and to take a sort of a stance in a couple of ways. Firstly, I think we need to get better information out there and to counter the misinformation. So for many years we've developed websites and resources, and one of the most recent we've developed is through the National Stem Cell Foundation of Australia, and there's this booklet you can pick up at the front or download
What
from the
you
website.
need to know about stem cell therapies.
And inside what we've tried to sort of do is break down some of this misinformation and some of the marketing claims, and to provide a guide effectively for people to think about some questions they might want to ask their doctor. A lot of the material we develop is recognising that you often feel isolated, that you may feel like you're making a decision on your own or with your family, but you also have a healthcare team around you, particularly here in Australia, where we have, a fabulous system of people you can turn to.
It's still very hard for people with cancer.
Absolutely.
You often feel like you're just a conglomeration of cells and organs, and that's what they're treating. They're not treating you as a whole person. And you also lose your agency in the system. You're there as a patient, as a bunch of cancer cells, you're receiving the treatment, hopefully it will kill those cancer cells. And so that's very tough. People they seek alternatives because there is a need for them to be treated as a whole person.
Absolutely.
So, I mean, in the stem cell arena, have you seen people with cancer seek out stem cell therapies?
I've certainly had, I receive a lot of inquiries from people with lots of different conditions, including cancer, and I certainly have seen advertising claims on websites that are for cancer treatment.
Yeah. And
cancer treatment for thymus and bone marrow or kidney or liver. And back to your point about sort of this sense of agency, a lot of us aren't prepared. If we get advice from our doctor where there is no other option for you, you often don't want to take that lying down. You want to do your research. As you mentioned, patients are often impressively well informed about the latest science. And I suspect Mark gets many of these questions in clinical practice. And we don't give medical advice as scientists, but what we try to do is reinforce those conversations with doctors like Mark, who really know what's going on and what is the best option for that patient right now, given their broader perspectives. But I take your point around the limited nature of that conversation and how people may feel very medicalised and not heard. And I think what we're seeing more and more of, and what I'd like to see more and more of, is opportunities to have a broader conversation. So we do have fantastic advocacy groups and patient support groups and other places you can turn to for information. I think the challenge is trying to work out who you want to listen to. So as I said, and I don't know if Mark cringes at this because it might make the consultations longer, but I think it's really great to go back to your treating specialist armed with your printout from the website or the fact sheet from the foundation and go and ask them for their advice and talk about it. And I think what we should be asking for more and more are those opportunities for conversations.
So, Mark, is this, you as a clinician-scientist, so you're right at the frontier of the research, but you're also working with patients regularly and who want to live and who want to have access to... Oh, is it working?
That one's correctly.
Oh, has it worked okay?
Yep.
Yep, okay. You're working at both within the clinic with patients and at the lab bench with scientists. Is there a tension there? Do you sense a sort of frustration that the science isn't hitting the clinic and impacting people's lives quick enough?
No, I don't,
You don't?
actually. I think, I really embrace a number of the things that Megan has just talked about. In fact, in my first consultation with a patient, I often meet them on what is the worst day of their life. Right? And so trying to give them information in small pieces that they can go away and action is really important. And I say to them, the first thing you're going to do when things start to settle down is hit Dr. Google, right? And I want you to do that because then you're going to come to me and ask me, are these things that I'm reading correct or wrong? Because I think it's really important as clinicians that we engender trust in our patients and make sure that they feel heard. And there is no question that is too silly to ask because this is their life and you want them to take responsibility for feeling like they are actively involved in every treatment decision. So I think the more we do that, the less likely charlatans are to thrive.
Do you encounter charlatans?
All the time.
What
All the
sort?
time.
What sorts of promises are they making?
Well, the ones that Megan has just outlined, and I think it particularly grates on me because they're preying on the desperate, right? And that feeding of false information gets perpetuated particularly when you hear bad news from conventional treatment. And it is in that which is the most vulnerable time that people get taken advantage of. And I think that's the part that really worries me the most in terms of the lack of regulation around that.
So you're a stem cell scientist as well.
Sure.
And so just let's explore the frontier that you're working at. Just maybe introduce people to the idea of what happens, what are stem cells and how are they relevant to the cancer story and why are you so interested in them?
Right. So, every tissue in our body, be it your skin, be it your gut, be it your blood, has at its origin a stem cell that populates that tissue. And that happens through normal development. And what we think is the origin of cancer is when a mutation in our DNA arises in the context of a stem cell that co-opts the properties of that cell, which is the properties for indefinite self-renewal, the properties for multipotent differentiation, the ability to differentiate into different cell types of that particular tissue. Though that confers on those
All the cells in our body originate from stem cells.
Correct.
And some differentiate into particular types of tissue types. And
That's right. But the one truth about cancer is that you can't get it without a mutation in your DNA. Right? So something has to corrupt the normal program of that cell in order for it to have all of the properties of a cancer cell, disrespecting its environment, growing out of control, moving to places that it was not meant to be. Right? Those are the properties of a cancer cell, but they often have features of the tissue-specific stem cell in which it likely arose.
And so what are you looking at?
Is how do we constrain that?
Yeah.
How do we constrain those properties of plasticity? What is plasticity? Is an ability to evolve very quickly, right? To meet a challenge without necessarily mutating your genome again. And a stem cell has this because inherent in its properties is the ability, if we take a blood stem cell, for instance, it can form trillions of cells through its lifetime. It was made to be able to produce cells for the duration that you are alive, 90, 100 years. And it can make every type of blood cell in your body. Now, when that becomes a cancer cell, inherent in that cancer cell is its ability to change shape and do whatever it takes to survive and meet whatever challenge there is. So how do we constrain that? That is probably one of the big questions that my lab is trying to answer.
Right. So stop those stem cells becoming cancerous.
Yeah.
Yeah. But you're also interested in the way in which sometimes a person can respond really well to treatment. Cancer cells are killed off. It's fantastic. But then the cancer comes back or it pops up in another part of the body. And so there's been, and it's not responsive to treatment anymore. The treatment that once worked no longer works. And that development of a resistance to treatment is a big issue, isn't it?
It is in every cancer, right? What you're describing there is a scenario whereby we see a patient, and often when patients present to us with their cancer that is untreated, sometimes they may have kilograms of cancer. Right? But when you treat that successfully, you can no longer detect it. You can no longer see it in a microscope or biopsying that tissue. You can no longer see it using sensitive techniques to try and isolate the abnormal parts of a cancer cell that have proteins on the cell surface, or even looking for specific mutations that were present in that cancer cell. But every technology used to look for those cells has a limit of detection. It doesn't mean that your cancer has gone away. I always try and explain to our patients that the first step to cure is remission. And remission means we can't detect it. Cure really reflects remission in time. The longer you're in remission, the more likely you are to be cured. But what we've learned over many, many years of doing this is that sometimes the cells go into this hibernating state. And they're able to stay in that state, undetected by our immune system, unperturbed by other challenges, and come back years, if not decades later.
Amazing.
And so that form of persistence, dormancy, quiescence, these are all of the terms used to describe these cells. That's something we need to understand if we're going to improve outcomes for patients with cancer.
This is possibly a silly question, but I don't believe any question is a silly question. But do cancer cells, those dormant cancer cells, do they learn from an assault from something like chemotherapy or radiation and modify themselves? And then when you try and treat them again, they are resistant to that treatment. Is there a plasticity in those cells?
There absolutely is. So if I give you the example, the type of cancers I treat are acute leukemias. And one of the most aggressive types of leukemias is acute myeloid leukemia. For 45 years, we've used basically the same treatment for that disease. Not because it's bad, it's actually quite good. We get 80% of our patients, up to 80% of our patients, into a remission. The difficult part is that remission is maintained in less than half of them. And sometimes that
over what time frame?
So majority of the people, that cancer comes back in the first 2 years.
Right.
But I've had patients where that cancer has come back, 20 years later. And it's really difficult to understand what controls that, because these cells are undetectable. So we have to build models to try and replicate that scenario, and that's where we have spent a lot of our time.
Yeah. With any success?
Certainly. I think, what we've understood is that you don't need another mutation in your genome for your cancer to go into this state, to be able to survive. In fact, it's probably easier to adapt by leveraging other things that are available to a cancer stem cell, changing the gene expression of that cell to adopt a new state that ignores the therapy that has just been impressed upon it and is able to adapt to that much more quickly.
I'm interested in what it takes to get fundamental science into the clinic. David Elliott, I'm interested in your impression of that. So you're right at this stage where you're testing drugs, possible drugs or possible substances on little stem cells in a Petri dish, and some seem to be responsive, some aren't. Okay, that's that stage. But what would it take for you to get that work into a child?
Yeah, I'm going
with leukemia.
I'm going to give you the answer I give my institute director. Yeah, money. So.
But what would you do with that money?
Yeah,
so just to give people a sense, because it's a great frustration for people. Well, why can't I get it now?
Sure. So I mean, it's in part a joke, but in part it's true. It takes I think it's around $1 billion to get a drug into it. You need an ecosystem, right? So you need clinicians, you need basic scientists, you need people who know about drugs, medicinal chemists, you need business people, you need, I think, the community support as well. We have a big program with, patient advisory groups that talk to us and tell us we need this now, and they...
Do you feel that pressure?
Yes, I do. I think that's why we're here, to help people. So, and so that's part of the drive. And then once you've built that team, you need regulatory specialists. You need to go to regulators. You need to get all of these hurdles ticked off, and you need the ecosystem, which is why, I'm sure Mark would agree and Megan would agree. We work internationally because you need not all the expertise you need is local. So our dream is, my dream is to have something in clinic before I retire. I can see retirement now, thankfully. And so how do we get there?
So say you found a candidate drug, what then?
It's for a drug, it's a well-worn path of preclinical animals, testing, does it work, and then into clinical trials. So we need a series of things to come through. There are larger, we use mice in the lab, but there are now, there are larger animals that we would use to do the preclinical work to go into humans, and then you're talking to clinical trial specialists about how many how many patients do I need to see to make sure this is effective, and at what dose, and all of those sort of things have to be put in place. So,
and do those clinical trials happen in Australia?
Yes, they do. We're I don't know. I'm not a specialist in this. We don't lead many of them, but we do. We're very well regarded as a place to do clinical trials because of our medical infrastructure, our Medicare numbers, record keeping, pharmaceutical benefit schemes. All of those sort of things are in place, and we're actually quite a good place to do clinical trials. I suspect many people running them just want to come out and have a visit to Queensland and go scuba diving on their way through, but that may be a bit cynical. But so it's a good place to do because of the professional, the medical system here, right? So they want to do it here. And so it'll be done collaboratively. I think, in the cancer space, I think the classic Australian one is probably a drug called venetoclax, which was a joint thing with the WEHI and the Walter and Eliza Hall Institute and AbbVie and I think Genentech, so a three-party thing, an international program, and now I think it was led out of the Peter Mac, the trial maybe. And isn't it now treating
that's
patients?
And what is it treating patients for?
I'm going to say B-cell lymphoma. Am I right, Mark?
It's a range of blood cancers.
Range of blood cancers. Yeah. So blood cancers in general. Yeah. So, and the discovery was actually probably made in the WEHI in the 1990s. So to give people a frame of reference, I think the gene that it sort of targets, or the gene family it targets, was discovered in the early 90s.
Right.
And so it came into clinical use 27.
So 30 years later.
Yes.
Yep.
Yeah.
I think so.
But that journey, if I could
jump in.
is, that journey, it's really important, whilst there's frustration associated in the lay media and lay press about how long it takes to get a discovery in the clinic, a discovery in the lab into the clinic. I guess it's important to outline what the principles are. Yeah. So once you make a discovery, you then need to make a drug that targets whatever you have found. That takes time because it has to have medicinal properties, which means it is tolerated, it is able to get into the cells in the body, etc. But once you've done your animal testing, the principles of a clinical trial are very important to stress. And they are what aligns with what we practice clinically. A doctor's job is to first do no harm. Okay? So the early clinical trials are to test not how good this drug is, but is it safe and is it tolerable? Right? And so that's your earliest clinical trial. As part of that, we inevitably see some patients respond better than others, and that guides us into where we might focus our attention on this drug. So that is the next phase of clinical trials. If that goes well, and each clinical trial is measured in years, right? So when I still worked at the University of Cambridge, we made a discovery into a protein that we built a drug with a pharmaceutical company. I started that clinical trial when I moved back to Australia in 2014. That was the early stage clinical trial. Is it safe and tolerable? That took 10 years to do, okay? Because you need to recruit patients, of different
And so
types,
is that drug in the marketplace now?
So that drug was proven to be safe and tolerable.
For what?
For a range of blood cancers. Now it needs to be combined with various other things for it to be efficacious.
So it's a slow game.
It is a slow game,
Yeah.
but it's important to understand that it is a slow game because we're trying to protect the interests of the patients taking that. The last thing we want to do is charge ahead and cause more harm than good.
Megan Munsey, you wanted to pick up on something, and I also would like to pick up with you that when you're a cancer patient, you are in a very vulnerable and desperate situation often. And I wonder whether that vulnerability makes you more susceptible for being a bit of a guinea pig in trials.
I think that's absolutely right. I think this sort of notion of try anything and this sense of frustration that perhaps there is something coming, but why do I have to wait? And if I can't get that, what else can I get? So I think managing that, as we've talked about, is really challenging. And I wanted to kind of come back because I'm really glad Mark raised that issue around safety, because I think that is foremost. And it's not just, obviously, it's about protecting people from harm, but it's also making sure our precious resources in terms of the health budget that we do have or can be utilised to support new and emerging technologies is being used appropriately. So we don't want to see money wasted and resources and effort wasted on something that doesn't have the return we want. So I think that's also a really interesting kind of fine line.
But waste
and
is an
balance.
interesting thing, isn't it? Because you don't know whether a drug's going to work or not in a trial or not.
And I think that's that.
Is it a waste to test it?
Definitely not a waste to test it. It's a waste if there are treatments that are still on the market that have been shown not to be effective. So we have to be nimble. And we have to keep monitoring. So Mark was just talking about the different phases of clinical trial. And I think it's worth mentioning that, we also monitor drugs when they're in the marketplace. And we should, if there are unexpected outcomes or if there is a better therapeutic that comes onto the market, perhaps an old drug is removed or an old therapy is removed and funding associated with that. That's what I was sort of implying. So I think that kind of understanding a little bit more about the regulatory system is really important. And it comes to comments I often get from people about their frustration that they feel like TGA or the FDA are the gatekeepers and are restricting access. And this sense that people feel powerless or even angry about that. And I think I'm always really curious to know more about how they see the regulations, because I think they are there to protect us. And I think that is something I want to see upheld.
Although there is a perception sometimes that drugs might be approved in the US and we're slow on the uptake, perhaps relative to the US. The FDA, the equivalent in the US, might approve a drug and then it takes time for the TGA here in Australia to go through the same process. Although who knows what's going to happen with the Food and Drug Authority in the US now under a Trump presidency. Mark Dawson, are we nimble enough with new cancer therapies?
I'm glad the question wasn't what's going to happen to the FDA under a Trump. Are we nimble enough? I really do believe we are. We, particularly in Australia, embrace this idea of making sure that we're doing the best available for our patients at the earliest time point. I'm very fortunate. I work at the Peter MacCallum Cancer Centre. I could unequivocally say, having worked at many places around the world, that this is one of the premier cancer centres, not just in Australia, in the world. And we have, an exceptional program of early drug development. If there is a discovery that is made that leads to a new drug, not infrequently, we at the Peter Mac are the place where the first patient in the world receives that drug. But that takes a certain amount of expertise. You need to have people with training and expertise in early drug development, knowing how to cater for unforeseen side effects. You need to have the infrastructure to be able to do that. So I don't suppose we'll ever come to a point where every hospital in Australia has that form of expertise, but I'd like to see every state in Australia have access to that form of expertise and experience. And that's where I think we need to make it more equitable across the country.
What would that take? Equity of access?
I think in many ways it takes an investment from our state and federal governments, understanding the urgent need to implement specialist centres in every state within the country that is able to execute this form of clinical trial management, and then training the people and disseminating that knowledge across the country.
Let's come to you for questions or comments. Yep, I'll come to you. I'll hold on to the mic. You don't need to hold the mic. And do we have any online? We do. Excellent. Thank you, live stream participants. So I should just remind you, if you're on the live stream, we'd love you to participate by sending your questions to, and I'll just say it all in one and then I'll spell it out, tanya@scienceinpublic.com.au. That's Tanya, Tanya, at scienceinpublic, all one word, .com.au. Email away and we'll get the questions through. And I'll come to you. Thank you.
David, I was wondering, with the chemo and its effect on the heart, is the percentages higher in children than adults of it affecting the heart?
That's a great question. The answer is no. It's the same. But, I like to, maybe at 65 it's okay. It's probably not okay at 5, right? These kids have got to go through puberty. They've got another round of sort of heart growth to go. So it's not that they're any particularly more sensitive because they're kids. Right? Adults too, particularly women getting breast cancer with a combined therapy, are also a lot more sensitive. So we have we make sure that our cell lines are female and male, and we do the check because women are much more sensitive. But it affects all equally. Yeah, It's a good question. It's not just because of they're young.
Coming to our live stream audience.
Great question for all of the panellists here from Nick. Given that the Medical Research Future Fund has recently noted consumer-driven research as one of their 12 priorities for 2024 to 26, how are the panel valuing and utilising consumers in their work?
Very good question. Go for it. David Elliott.
Sure. We have patient advisor groups for every program we run. Have done for maybe 5 or 6 years.
So what do they do? How do they change what you do?
So our Children's Heart Tissue Bank patient advisory group, for example, they are kids who've had heart surgery, or their parents, or young adults who've survived heart surgery in various guises, and we present to them monthly. They tell us what problems they faced. They are really interested in the science, and I think really what they do is just keep you honest, right? So this is the community you're serving. It's at the sharp edge. I mean, Mark is a bit different. He sees patients, and Megan and I. Megan probably sees a lot of patients actually, but for us it's important, and I think it's really important for our group, our young researchers, to be exposed to that. They're not just sitting in the lab under some fluorescent lights, moving clear liquid from one tube to the other.
Plenty of that going on though too. Megan Munsey.
Yeah, I was just going to say that in addition to what Dave was describing around this with patient advocacy, these formal roles, we also try to engage in many other ways. So we do hold Meet the Scientist sessions, things like that, public forums like this, and we are responsive to emails. I think that's really important. I'm sure Dave's had this experience, and probably Mark, when there's a story about their work in the paper, people will contact them. And we really do try to respond and to contextualise what's happening in the lab to that inquiry. So, we do try really hard to involve patients and patient advocacy groups throughout all of our work.
Do you have any thoughts about that, Mark?
Look, just to say that it's incredibly important to have consumers involved in research. I think you've got to think about the research spectrum being from basic discovery science all the way through to implementation, and consumers can contribute along that entire spectrum. So I think it's an incredibly important part to play.
Thank you very much for this wonderful discussion. And Megan, a question for you, because I think it seems like you have the greatest interface with the broadest lay community. What does the lay community think stem cell therapy is?
So it's a
Are
fantastic...
you a stem cell scientist?
I'm a clinician, researcher, dabble in the stem cell piece.
Are you coming to the conference?
I'm the director of Segenzi, so
Oh, thank you for providing this beautiful facility. So I think that's a really interesting question and an important question to say, because I think in the community, when people when they get the diagnosis that Mark talked about, the news that they don't want to hear, one of the first things, no matter what that diagnosis is, whether it's cancer, whether it's Parkinson's, whether it's diabetes, they'll go online and if they don't know about stem cells already, one of their friends or family members will raise it. Isn't there a stem cell treatment for that? And so that's something that we're, acutely aware of. I also think the other thing that gets lost in translation very much is, what are we talking about? So Dave's doing research on models where he's taking stem cells or cells from a child, making stem cells, then making heart cells from those stem cells and trying to understand more about that interaction with drugs. So we do a lot of modelling, but we also take stem cells to try to make a cell therapy. And we do that for a lot of different conditions. Explaining that in a sentence can be quite difficult. So I think we're always trying, like back to the previous question where we were talking about patient engagement, we're trying to tailor the information, the message that's relevant for that condition at this time, and try to avoid extrapolation. So back to the kind of concerning practices that I've been monitoring now for almost 2 decades, it's very easy, it's a very positive message to just say stem cells are fantastic, which of course we think they are, but stem cells can cure all, or can help cure all.
So you've also seen in the literature cases of people who have had end up using stem cell therapies from private clinics, unproven, and they end up with cancer because of it.
Yes, that's absolutely true. So there's a very graphic case, and unfortunately, or understandably, people who have these experiences don't often speak out, Natasha. So in this rare example I'm thinking of is a gentleman called Jim Gass, who his case was written up in the medical literature, but he also did an interview with the New York Times. And it's quite a chilling read.
What happened to him?
So he was a successful businessman, an American guy who had a stroke, and he'd done all of his rehabilitation, but he had some deficits still, some physical deficit, and he was frustrated. He thought, surely there's something better out there for me. So he did his own research. I'm not quite sure who he consulted in terms of his medical team, but he did visit 3 clinics around the world because he had the money to do so, and they offered all sorts of different stuff. And
so-called stem cell therapies.
Absolutely. He thought he was getting stem cell therapies. And I suspect he thought he was getting legitimate stem cell therapies. And one of the interventions involved injecting something. We think it might have been fetal stem cells. We're not quite sure what it was, but into his spine. Because, of course, the theory was that if we get more nerves or stem cells to make nerves, that will help with stroke. From my perspective, it's pretty weak, that argument. But for Jim, he thought it was worth a try. So he had it. And then what happened was some time later, he developed some pain, he developed paralysis, and he was rendered a quadriplegic by an aggressive tumour that formed from what we think of that donation. But
Which is a possibility when you inject stem cells, fetal stem cells into someone.
And if you don't have the recipe right,
they can turn into a tumour.
Or even if you took cells that you've cultured, skin cells, for example, you've cultured for a long period of time, they can pick up a mutation like Mark was talking about and change their fate. So in that case, Jim was very keen to share his story, as was his family. So it's really a gripping and really, I think, key example of what can go wrong. So you can have these you can spend a lot of money, So there can be financial harm, there can be physical harm. But I often think also about the psychological harm. The fact that, again, we all think about stem cell therapy as being helpful. And if it hasn't worked for you, it may make people who've paid for these alleged treatments feel like there's something wrong with them. That why hasn't it worked for me? If this company have already treated, as they claim on their website, 10,000 people, and obviously they were happy, because I've never heard a bad story. So if it hasn't worked for me, I must be at fault. So there's a kind of this psychological manipulation as well that obviously I quite object to.
So we've got better at regulating those clinics in Australia, haven't we?
Yeah, it's a really good news story.
Not least because of your advocacy around this issue. We're now legislating.
Yeah.
But there's still the issue of stem cell tourism as well, so people flying overseas to other countries.
That's right. So even though you can't get stem cell, even though there's an explicit ban now on advertising.
In Australia.
That's right. The TGA now enforce this ban.
On advertising, but what about the
advertising?
actual existence of the clinics?
So we can still see a couple of recalcitrant clinics, but it's very much contracted. So from over 50 clinics, there's now less than a handful, so less than 5. Some of them are continuing to, I think, perhaps exploit a small loophole around a hospital exemption. But I do think they should not be advertising, and of course we bring this to the attention of the TGA all the time. However, if you're someone who is doing your research online, it doesn't stop with the Australian border. You might find a clinic in India or China or Mexico.
Or America.
Or America. And you might just think, I want to give it a go. So it's I think, whilst we've had success, and I think it's important that we keep watching the field, and I'm always very happy to hear about concerning practices people see, because through that kind of monitoring and observing what's happening, we can bring it to the attention of TGA and we can make a difference. So it's challenging, but I think twice is my message. If you see something that sounds too good to be true, think twice and talk to your specialist.
Thank you so much, Megan. Has anyone had experience of cancer treatment and felt frustration about the kind of, well, look, I want to come to you. Do you mind if I do? Has anything that's come up today resonated with your experience?
Yes.
I've got the mic.
It has. I think, Mark, if I can just put this to you, some 4 years ago I had a little thing taken out of my ear, and obviously it was a minor case of cancer. Some 6 weeks later I had a little pimple behind my ear, and went to my local quack, and he said, don't worry about it, antibiotic will fix that. Sorry about my mouth, I'll come to that in a moment. Anyway, it grew to about a 10-cent piece in a week. I said, well, take a sample. He said, no, mate. He said, you're in the hospital, we're going to have that removed. So about a week later, 12 and a half hours under the mic, they had it under the knife. They finally got rid of it all, taken it all out around, everything was removed. Had radiotherapy, and I was okay. I'm a bit tired in the morning, that was okay. And I thought I had it all until about another six months later. I said, my tongue's not feeling really good. So I had every test done possible, and they all assured me I was okay. I said, I'm not okay, I can't eat, I can't drink properly. And I was told at the head of radiotherapy I had thrush in my tongue. I said, I'm sorry. I had 3 doctors look at it, I said, you're okay. Finally, my master doctor came in and he said, sorry to tell you this, mate, you've got cancer in your tongue. Not a very nice thing to go through. The operation was fine, but the radiotherapy for that, up here in the head, kills you. I've had more tubes in my body than you care to count. But what it does mentally, it shocks you. I was lucky, I had a good hospital and a lovely wife that looked after me.
Yeah.
I appreciate that.
Thank you so much for sharing that story. I appreciate it. And I know others who have had throat and tongue cancer, and it's a really tough diagnosis, isn't it? And the screening and scans. And this is the challenge, isn't it? That it's such a raw experience for people, isn't it, Mark? And you're dealing with this grief every day while also trying to work in a lab as well.
Yeah. That's a very courageous story. I guess, if I kind of think of what you did very well throughout that, was to continue to advocate for yourself, and continue to tell people that I don't feel right and there's something wrong here. You need to look harder. And I can't emphasise how important that is in the patient journey for you to continue to advocate for yourself. Because
It's a tough ask though, isn't it?
it is a tough ask, but you know yourself when you don't feel right. And that's unquantifiable sometimes for a medical specialist. They will do the tests that are possible, but as we talked about before, those tests are only as good as the limit of detection. So the assurances that you were getting given were probably because the test was not good enough at finding this. But your persistence has helped in many ways find you a solution and likely saved your life.
But it's a raw experience, isn't it?
Of course,
and
of course.
When I talk to people who have survived cancer, it's often the psychological traumas of the experience that live on, even if the cancer cells are no longer in your body.
Of course.
Thank you for sharing that story. Tanya.
My sister had breast cancer a number of years ago, and the story that we heard before, there's a reason why they call aggressive therapy, and I know that my sister was really frightened of what was ahead of her. The converse is that chemotherapy, radiotherapy, surgery all sound scary. Goji berries and supplements and this, that and the other thing sound a lot less scary. And my big fear was, or my need was to protect my sister from well-meaning but not well-informed friends on Facebook. What advice, Mark, do you give patients, or Megan, what do you advocate for so that we can protect people from I guess you'd call it the competition, the alternative that is offering hope without the evidence, in the face of cancer treatment being very scary, very frightening?
It's an important question, isn't it? Because people also need to feel like they are somewhat empowered and have some agency to treat other aspects of their body and mind during a cancer experience, don't they?
Absolutely. And I think, the single most important thing as a clinician you can do at that stage is to build trust, and be very open about what information you're giving, what questions the patient should go and seek information on, come back and ask for reassurance where it's possible, and just to be involved in that journey, and to continue to reassure that in this country, we're so blessed because there is nothing that restrains us from giving the world's best quality of care. That's not exclusive to the people who can afford it. In here, we will treat a prince the same as the way we would treat a refugee. And that's not true everywhere in the world, And so it's really emphasising that they are going to get the best quality of care. And whilst there's absolutely uncertainty throughout this process, these treatments are aggressive. And I think, at least in my experience, what I've found is being open and honest about the toxicities that are likely to be endured during this treatment helps prepare the patient for what the journey is ahead, and further engenders that trust where they feel comfortable coming back and asking you about questions.
Megan, did you want to add to that?
Yeah, I just wanted to say that I completely agree with everything Mark just said, and just also put a shout out there for all the amazing community groups that are there who are very aware of these sort of deviations from the norm, and who can provide great information and perhaps back up and provide additional context to what their clinician has raised. And I'd also say that not everyone's as fortunate enough to have a clinician or to as great as Mark, and perhaps to have that relationship. And you can go and get a second opinion. You can look, as Mark said, you can exercise your agency through trying to find the right doctor for you.
I spoke to a fellow recently at an event who had sacked his oncologist. His needs weren't being met. and the clinician didn't have the right communication style for him and wasn't seeing him as a whole person, was just treating him like a bag of organs, didn't suit him.
I couldn't emphasise that point more. You know, it's your life. You know, you don't need to worry about if you're going to hurt this person's feelings. If you can't find rapport with this person, if you can't feel like you can trust them, seek another opinion.
It's scary for people though, because
Of
once
course,
you're on
of
track,
course, of
you've got to
course.
feel like you've got to start again, don't you?
Of course.
And David, I mean, you're right there looking at those stem cells in the dish, but have you found that working on the issue of childhood leukemia has connected you to a patient group?
Yeah, definitely. Much more engaged with, you know, I work closely with a haematologist, Rachel Conyers, in the hospital, and we see some of their kids, and we can follow those journeys. So yeah, it definitely does, and I think there's an added element in the kids' hospital is the whole family's involved, right? So there's the stress of the parents. You know, there's a major treatment of the children. There's stress on the whole family, and I think one of the, as Mark's touched on several times, that the Royal Children's Hospital is an amazing place as well. You know, it's just down the hill.
This
from
is
the
in Melbourne.
Peter Mac in Melbourne, and you know, Adelaide has another children's hospital as well. Similar, multiple disciplines looking after you. You know, if the parents need some counselling, they can get some counselling. So that that access to this amazing healthcare system is very important.
But sometimes the disciplines are very siloed. I think people I know who have had cancer have gone, well, they treat that bit of me and they treat that bit of me. Can they all please just talk to each other? Megan, you had a comment, and I want to come to this question.
Yeah, I just wanted to go back to the fact that we're also encouraging and also enabling young scientists, as Dave mentioned, to meet patients. We want to move away from the model that researchers are working on a condition where they're yet to meet a patient that is suffering from that condition. So I think acknowledging, listening, learning from lived experience can be really invaluable for the research community as well.
Absolutely. Hi.
Hi, touches on well. I'm Sagency Consumer Advisory Group. It's really good. Unfortunately, the public hospital system, at least in Adelaide, doesn't quite work how we don't get a choice. You know, I know personally my father, it's taken 10 months just to get from a referral from a GP to actually see someone in public hospital for aggressive prostate cancer, then was in lymph nodes. So it's not quite ideal. You can't sack and get a second opinion. You take what you get, unfortunately. My question was actually, and perhaps oversimplified, so forgive me, I'm not a scientist, but is there a reason why we do not, in aims of making treatment worth it in terms of less symptomatics, things for the patient, why don't we look at basic genetic studies in terms of how we metabolise drugs, knowing some patients are obviously more sensitive, tamoxifen being a really good example?
Thank you.
Okay. Yeah, so that is a great question. And you know, what you've just described is called pharmacogenomics. So that's where a given individual on their genetic makeup, you know, how do they deal with a particular drug? And I can say that we're starting to implement it here now. So some services are coming, and you know, but you know.
So what would that look like from the patient's experience, and from a GP's experience,
so from a GP or
specialist?
So the system that we're sort of trying to set up is that you would take some blood, patients will be sequenced, then a report goes to the specialist. This patient will handle this drug poorly, this channel will handle it, so they'll get an
So their
automated
genome
report
is
production.
sequenced.
Genome is sequenced, so some blood.
Variants are looked for.
Exactly. So,
that confer a better
Confer
outcome for
sensitive
that
or-
drug than that drug,
Correct,
etc.
correct. So
Yeah.
there's some, you know, there's a few that we have a handle on. There's many, many more that we have no idea of. And so there's, you know, what are referred to as clinically actionable variants, which is just a small change in your DNA that can inform what choice of medicine you're given. There's not that many, but there are some, and we're trying to implement those now. So we're moving past research stage into implementation stage. And the implementation stage for the patient should look very simple. You know, a little bit of blood, it disappears to a sequencing clinic, and then a report lands on the specialist desk, on the GP's desk, and in your inbox too, should you choose to to tick that box.
And but the challenge for pharmacogenomics has been also keeping the medical profession up to date with the latest
Correct, so
information and the
yep.
latest data, and how do you translate that into clinics? Mark?
For many drugs that we give our cancer patients, there are known gene variants that cause you not to metabolize that drug well, and therefore end up on the extreme end of the toxicity scale. And so for many of these, from decades of research, we now prospectively test our patients before giving them that drug. Right? So what pharmacogenomics ultimately feeds into is informing us on how best to use some of these drugs.
Yeah, we've got one more question, I think. Thank you. I'll just dash up. Keeping me fit. I can't really pull off the Oprah thing.
So talking about new technologies, and also we're reflecting on the fact that we have radiation and chemotherapy for a long time. What excites you in terms of research? What is coming? What is the new technology that we have to look for in terms of cancer treatments?
Awesome wrap question, thank you. Who'd like to answer?
I'll have a crack first, I think. So I say to everybody who walks into my lab, and in my lab, I have both a mix of clinically trained scientists as well as, basic discovery scientists. I think what's an important core aspect that I want them all to embrace is curiosity. Because without curiosity, there are no discoveries. And without those curiosity-based discoveries, we've basically accepted this is as good as it's going to get. So how do you get new cures? To think outside the box and generate that, right? That takes a lot of time and effort. But the innovations in technologies right now has enabled us, we've gone from being able to sequence the cancer genome to now being able to see this at single cell resolution, to understand this variability that drives the way different cancer cells adapt, metastasize, spread, et cetera, et cetera.
Because you're tracking single cells across time, is that right?
Correct. Right. And so I think our understanding is in cancer biology is embracing advances not just in biomedical research, but in physics, chemistry, and other disciplines, bringing those together to really see a cancer at unprecedented resolution. And so I'm very excited by what we're likely to discover in the next 5 to 10 years and what that means for the next generation of treatments and how that will change the natural history of many of these cancers.
Amazing. Getting really close and intimate with cancer, on its case, well and truly. Any thoughts?
So I agree. It's technology that's pretty exciting, and we just live in an amazing time of the array of tools that we can apply to our problems now. And that's, from physics, from maths, from AI, we just did what we would call a drug screen in silico where we looked at 14 trillion combinations, right? Well, I didn't, someone who's young.
And you using AI to crunch that data?
Yeah.
Big data sets.
So that allows you to get more and more resolution on what the problem is, and then that allows you to get a really targeted therapy. One of the common drugs, as we've all said, has been in use for 50 years, okay? But it's pretty broad spectrum. It hits everything. So now the progress that we're making means you can get a much more targeted therapy and then use different delivery systems to get it into cells.
Could you imagine a time where it's not chemo, it's hyper-targeted chemo? What would that look like, Mark?
We don't have to imagine a time. It happens now.
It happens now.
Yeah.
So how, is it delivered in a different way, or is it right there, you sit down and you cop it sweet across every body organ?
So I think about a cancer that I treat called acute promyelocytic leukemia. This, 20 years ago, was an invariably fatal disease. Right? It is now treated without chemotherapy. And it is treated without chemotherapy because we understood this cancer at the molecular level. And we give targeted therapies that are specific for this cancer that cures now 95% of these patients. So that, that's what the power of scientific discovery can do. And there absolutely, I think we should all embrace a time where, indiscriminate chemotherapy might be a thing of the past. But that's not a vision for tomorrow, it's a vision for 15, 20 years' time.
You're also developing epigenetic therapies. So what are they, briefly?
Well, if we think about the fact that every one of us in this room has trillions of cells, and if we stretch out the DNA content of that cell, for any human being, you can walk to the sun and back 600 times. Right? Now that DNA needs to be repaired, it needs to be replicated, and it needs to be expressed as our genes. What epigenetics is the study of that regulation that enables the machinery to access the DNA at the right place at the right time to specify to that cell, you need to be a blood cell, you need to be a liver cell, and you need to be a skin cell. All of those cells have exactly the same DNA content, the same instruction manual. It's just not every page is being read.
I think of it as sort of the machinery that surrounds the DNA that switches genes on and off by blocking their action in some way so that they can't, encode proteins in the body. Yeah. So what are epigenetic therapies?
So epigenetic therapies are not going to cure anyone by themselves. What they are going to do is constrain that plasticity that I talked about before, limit the number of options. You can't turn the page to read a new manual, right?
So switch off key genes that are involved in cancer proliferation.
That's right.
Yeah, right. Megan Munsey, final comment to you.
Well, I was reflecting on Mark's use of the word curious, and I think I want to see people remain curious about medical research. But again, as I said earlier, I think how you find information and the information that you find, how you then process that, I think we've got to think about a bit more deeply. So it's going beyond sort of face value, going beyond the simple ad. And I think that Dave mentioned AI and modern technologies. And in my world, in terms of communication, there are better ways to get information out there. As an older person, I'm not so keen on TikTok. And when someone first suggested I put a chatbot on my website for patient information, I was kind of horrified. But I can see that we're not fully exercising, fully utilising modern communication technology.
The philosopher Peter Singer, the bioethicist, he's just had a chatbot created that's been trained on his whole corpus of work. And so because he gets so many questions from people, because he's quite controversial in some of his interrogation of ethics, he now just gets them to trial this chatbot and give an answer to a question that they put in. So there you go.
There you go.
He thinks, he's getting older now, and he says, well, look, this will outlive me.
Yeah, so again, it's another use of AI that perhaps we shouldn't dismiss, we should embrace and think about how we can use it well, which is really about science.
Thank you so much. Thank you. Oh, you just squeeze one more in really quickly and then we'll call it quits. Thank you.
Hi everyone, thank you for your information and sharing today. I understand that after the cancer treatment, that would be really challenging for customer. And I would like to ask, is there any other approach comparing to other injecting stem cell, like you can take any nutritional or prefer healthy diet, especially that's the one that kind of like known as providing enough phytonutrients or antioxidants that can protect your cells, nourish and repair from all the oxidative stress from the environment. Is there any recommendation or any research into that you can recommend for those who has suffered from cancer already?
It's a very important question because a lot of people who have cancer go, well, what can I do? What can I do physically, or what can I eat? Mark?
So there's virtually no information that supports the evidence that taking vitamin supplements prevents or improves your outcomes from cancer. Right? What I tell my patients is to try and embrace a balanced and healthy diet. And not infrequently, when they are in the hospital, they see a dietitian. And the dietitian is the most informed person to be able to give you that advice on exactly what you should consume and what are, the right foods for you at that particular time. But I think
It's frustrating though. People would like to see more research done around nutrition and cancer, and it's not. There doesn't seem to be a lot there.
that's not to say that there is not research done. There's a whole body of work done on how your body's metabolism influences the cancer journey. What I guess I'm trying to particularly impress is that there's no evidence to suggest that supplemental vitamins will change the natural history of this.
Which is contra to many of those wellbeing influencers on social media who see food as medicine. But it's a very potent discussion, so I'd like to see more research done on nutrition and cancer. Did you want to add anything, David? No? We're ready to rock? Hey, thank you so much for being here on a Sunday afternoon. It's a beautiful day out there. I hope you enjoy the rest of your afternoon. We did record today for Big Ideas, which you can find on the ABC Listen app, or nightly, Mondays to Thursdays at 8 p.m. on Radio National. That's 7 to 9 a.m. here in Adelaide, or across Australia indeed, on a whole range of different frequencies. And I'd love you to thank Professor Mark Dawson, Associate Professor David Elliott and Professor Megan Munsey. And I hope if you're attending the conference this week, have a great week. Thanks for being here. Enjoy the rest of your day. And thanks to all the supporters of this event, the National Stem Cell Foundation of Australia, the University of Adelaide and their Immunogenomics Cancer Institute right here. Thank you for having us in your building. And also the Australasian Society of Stem Cell Research, who's meeting this week in this beautiful city of yours. Go well. Thank you.